Immortal, Thin, and Immune. How Far Are We? By Keenan Maley. To be human, is it our heart, our soul, our body? Is a man deemed as such because of the blood in his veins, the meat on his bones, and the brain in his skull? Or is a man gauged on the actions he makes, the personality that shapes his being, and the consciousness that allows cognitive life? This is a conundrum faced by those who study biotechnology every day, particularly those involved in the field of organ transplantation. Organ transplantation is a quickly growing science in the field of biotechnology, with new discoveries found with startling frequency. From functioning lymph nodes implanted and grown within the hypothalamus to the way of ward off the aging process with a simple injection, organ transplantation is clearly one of the most interesting fields in biotechnology. Is a man not entitled to a full lease on life? With the advancement in organ transplantation, people are able to have just that and perhaps even more, but there is controversy about the ethics on using such advancements. There are those who believe that the body is sacred and should not be tampered with, neither positively nor negatively. Those opposed are unfortunately primarily in the majority, and they believe that our bodies are already formed to be as perfect as we needed to be, and we as a species should not perform such hubris as to manipulate what we were born with. This type of thinking is detrimental to the advancement of scientific achievement and, in fact, intellectual pursuits in many other categories, even those unrelated to science. Humans as a species are strivers. They're never satisfied to be where they are at, always wanting to travel farther, faster, jump higher, longer. Living life to the absolute fullest is a pipe dream that everyone in the world wants to achieve. Whether fullest refers to his monetary worth or life experiences, quality or quantity is irrelevant. Living life truly to the human limits is not satisfactory. Laboratory mice are vital to the process of science. They are used as human substitutes where human testing is either too dangerous or too uncertain. Mice are the unspoken heroes that allow the progression of science to continue by sacrificing their health and well-being for the benefit of the human race. Any research that can improve the quality of these mice will inherently increase the quality of all research that involves the use of said mice. This is where new research that can increase the lifespans of mice that have been bred to age too quickly can find new life. Perhaps this technology can even be used to lengthen the lifespan of humans in the future. The disease that causes mice to age at an accelerated rate is called progeria. Progeria is a genetic disorder that simulates accelerated aging. The mice lose significant muscle mass, particularly in their hind legs, hunch over, and move laboriously. The same signs can be seen in naturally aging mice, even down at the cellular level while cells begin to lose their refinement. Progeria's effect on the body of a mouse can be seen as both a blessing and a curse. Due to the accelerated aging, the mice can show symptoms of side effects much quicker than the mice not afflicted with this disease, and scientists can experiment on a wide range of ages without waiting several years for the mice to age naturally. However, because it is a genetic disorder, it is passed down to its children. So if a scientist wants a mouse without progeria, they cannot use mice with progeria or the offspring of mice with progeria. This genetic disorder can be combated, however, by injecting the progenitor cells of healthy young mice into the old infirm mice. Their lifespans are lengthened by over 100% and even as much as 200%. The new cells can even direct the old and decrepit cells to correct their dysfunction. The idea behind this is that as the mice age, a variety of complications arise within their bodies. These complications include, but are not limited to, stem cell dysfunction or poor replication and differentiation of cells. The injection of progenitor cells slow this degradation of the natural function of the mouse cells. From this knowledge, a conclusion can be drawn that stem cell dysfunction is a major factor, perhaps even the greatest factor in aging. Through the creation and transplantation of artificial progenitor cells, scientists can replicate these results in both mice and potentially humans. The uses in mice could be very useful for science. Mice near the end of their life, but not the end of experiment, can be temporarily kept alive by rewinding its internal clock. The process used to synthesize the artificial progenitor cells could be replicated for humans, which can result in successful human trials and eventually a stable product available for sale. Though human study is still far off, the potential still exists to translate this research for human use. The possibilities on the beauty market are extremely lucrative, let alone the pharmaceutical market. People would pay top dollar to relive their youth or even just buy some time on this earth. Even those who are simply trying to prevent the aches and pains associated with old age, regain some mobility in their twilight years, or just not be bedridden in old age. The epidemic of obesity is a plague in developed countries in the modern world. This statement is perhaps the truest in the United States, where obesity runs rampant and those afflicted do little to change the fact. Americans as a whole have a love affair with fast and processed foods with an unhealthy disdain for exercise. These unhealthy practices could be connected to the leptin levels in the bloodstreams of those who perform them. Leptin is essentially the voice in the human body that alerts the brain to tell the body to stop eating. More specifically, leptin is synthesized as a person eats and is released into the bloodstream. As the concentration of leptin rises, neurons in the hypothalamus section of the brain take notice and send a signal to the frontal lobe to stop eating. Unfortunately, there exists a genetic disorder that creates abnormal leptin receptors that are slow at receiving and relaying messages that the body is full, and as a result, the person eats much more than is necessary. While eating more than is calorically necessary may seem innocent enough, when one does it on a regular or even habitual basis, the ramifications begin to build and snowball out of control. In laboratory mice, the difference is stark. Mice with abnormal leptin receptors weigh up to twice as much as healthy mice. A lack of a proper leptin receptor in the brain could be the mythical genetic obesity that critics have previously claimed non-existence, though those afflicted with obesity have tenaciously defended the theory. Thankfully, people with abnormal receptors have hope in the form of transplantation. The transplant entails that the patient receive an injection of immature neurons and progenitors that build a new leptin receptor in the hypothalamus. The idea behind the procedure is that the immature cells simply build what they were born to build, a healthy and functional leptin receptor at the site of the injection, the hypothalamus. The experiment behind this new concept is quite basic in theory. Before injection, the cells are marked with a fluorescent protein that allows the scientists to monitor the cells throughout the experiment. Then, the scientists use advanced high-frequency imaging to very accurately locate the target injection site with dimensions no larger than 400 micrometers. The scientists also use a pipette with a tip only slightly larger than the cell itself for extreme accuracy. After injection, the scientists conducted observations for 20 weeks. They saw that almost half the cells transformed into functioning neurons, mimicking those that grow naturally. With some intensive analyzing, the scientists were also able to determine that the entire range of neurons and proteins seen in a naturally healthy leptin receptor were created by the transplanted cells. The most impressive piece of evidence, however, is that the mice that received the transplant returned to a healthy weight, body metabolic rate, and body mass index. The transplantation of organs is often a very tricky task. The doctor must juggle several variables, not all of which are foreseeable. For example, even if the patient and the donor have the same blood type, an antibody left over in the transplanted tissue could cause an immunoresponse in the host's new body. However, organ transplants are still in their fledgling state, and as a result, scientists are finding new and exciting ways to improve the success rates and opportunities for saving lives. One of these new technological creations is the concept of using T cells to neutralize a deadly immunoresponse to the transplantation of a donor's organ or organs. The idea behind this is that T cells in a healthy individual bodies fend off immunoresponses of a similar nature to those found in the transplant patients. Therefore, scientists conjecture that if T cells that recognize the transplanted organ are within the body of a patient, the T cells will prevent a fatal response to the foreign organ. This theory has been proven to be true in preliminary tests of laboratory mice. When treated therapeutically with T cells, the mice were able to attain long-term protection against the primary complications associated with organ transplantation, namely inflammation and rejection of the transplanted organ. This is desirable of the generic immunosuppressive drugs available today because of the side effects associated with these drugs. Due to their blanket approach to immunosuppression, some immunoresponses that are desirable are unable to effectively fend off infection. This leads to a greater danger in traditionally innocuous diseases such as pneumonia and influenza. Also, these drugs only offer a very temporary reprieve of immunosuppression, requiring frequent doses to keep the effect active. T-cell injection, on the other hand, improves on the drugs in nearly every fashion. They are very specific in their immunosuppression, only preventing the response to the benign transplanted organ. They last a very long time, allowing for infrequent doses and long-term protection. In fact, T-cell therapy even promotes tolerance to the transplanted organs, potentially eliminating the need for therapy at all. Despite not having human trials as of yet, the evidence is firmly in the favor of beginning preliminary tests. Bone marrow transplant has created a precedent supporting the safety and efficacy of T-cell therapy. Though the tests appear to be safe, they should begin in small and focus slowly on increasing the doses to discover the ideal volume for each of the patient across the severity gradient. An experiment has been proposed by J. Mulcell Bile that is a three-stage affair. The first stage is a clinical protocol based on T-cell immune suppressive regimen. The focus of the experiment is to find a balance between the safeties of the volume injection with the effectiveness of the T-cell immunosuppression. The second stage of the experiment is to produce the T cells in a controlled environment to analyze the effectiveness of fabricating T cells for the purpose of therapeutic injection. Third and final stage is to create a comprehensive monitoring system in order to effectively and efficiently keep tabs on the patients and, as a result, respond swiftly and competently to any sort of complication that may arise from the trials. Regardless of the form of the experiment, it is vital to execute it professionally and prudently. This is a new technology and must be treated with extreme care. These experiments will pave the way for new and more elaborate T-cell therapies. Steps 3 and 4 cannot happen if the concept is stopped at step 1. It is important to remember that organ transplantation is still a young field of science. Though it is still young, it is very promising with its growth. And it is perhaps even more important to remember who we are doing this for, and that every man deserves to live his life to the fullest, whether that means he be elevated to new heights or return to a level to which he is accustomed. Regardless of whether or not a man has a prosthetic limb, heart, or brain, he is still just that, a man. A man who has the right to live in a world where he is not bedridden or chained down by a crippling disease or injury. The right to not be judged no matter the circumstances at birth. These are certain inalienable rights that organ transplantation can provide for those afflicted with such a curse as to not be able to receive the benefits these rights ensure.